Well, I hit the wrong button. Well, that's how it goes. Hello, everyone. I am Joe the Science Bro, and this is what we have started calling Science Talk. Over the past few weeks, we've been having discussions about different scientific topics. And while we did start with Alzheimer's, today we're going to kind of swerve in a completely different direction. Hello, everybody. It's good to see you. Thank you. I'm glad that you're excited for some science talk. I would like to take a moment to thank Dr. Balin, but I would also like to thank our guest today, who graciously is offering some of his time. And again, this is at the end of our day, so he's actually offering his personal time with this, and I am very, very grateful to him for doing so. Um, uh, our guest today is Dr. Arturo Bravo Nuevo, and I hope I said that right. Please tell me I said that right. Um,
You
I
did.
have always referred to Dr. Bravo as Dr. Bravo for short. Um, but I am also very glad that he can be here with us. Dr. Bravo, if you could for a few moments, just tell us what it is that you currently research in science. You don't have to give us any specifics because we do understand the necessity for certain things to kind of remain in house.
Well, first of all, uh, good evening, everybody. Thank you, Joe, for organizing this. I think this is a great idea, and I think we were chatting about this before. I really appreciate you making an effort to get the science out there. And I think one of the problems that scientists have these days is we're not very good at communicating the science. Um, you are a great communicator. I've noticed that in the past, and I really appreciate the invite, and I appreciate all the people that are here listening. Also on their own free time too. So thank you. What we work in the lab. So I have a lab at PCOM, same place as Joe works in, and I've been there for 6 years. Most of our work relates to um, diseases of the eye. Um, and most of them, although not all of them, to the retina. We have done a little bit of work in the brain, just because the retina is formed by neurons, and so is the brain. And I'm excited to talk about it in a little bit. But we were mostly on neuroprotection. What can we do, not to cure a disease that is going to leave somebody blind, but to slow it down? I think it's a very more realistic approach. Uh, a lot of people are just trying to cure a disease, but for us, it's to slow it down. So, somebody,
Mm.
for instance, who has retinitis pigmentosa is gonna start by the time they're 12 or 13. They're gonna have start having problems when they try to go to the bathroom at night, because affects mostly the rods, which are the cells that detect are responsible for vision at low light. When you have like at night, when you go under the stars, those that kind of light conditions, or when you go to the bathroom and you don't have any lights on. Um, so those kids will start having problems going to the bathroom. They get lost, they bump into things,
Mm.
and then by the time they're 20 something or 30, they might be completely blind. We're trying to delay that progression, and so they are able to have vision until their 50s, and be able to, that might be the difference in some people in being able to see the faces of the kids,
Mm.
or never being able to see the faces of the kids. So we work in a series of diseases. A lot of our work is in that neuroprotection, but we also work in corneal transplant. Why do corneal transplants get rejected? When you're gonna give uh, tissue for donate a tissue to save somebody's life.
Mm.
Usually, you have to make sure that there's immunocompatibility between the donor and the recipient. But that's not the case in the cornea or in the lens, because those don't have any blood supply usually. so it shouldn't be immunocompromised. But still, um, 2 out of every 10 corneal transplants fail,
Mm,
and people don't understand why. Yeah. So we work in that. We work in glaucoma. Uh, as I say, we work in acute models of or reproducible models of Alzheimer's and
mm-hmm,
neuronal plaques. We've done a little bit of work on that. So we work in diseases that affect newborn kids, retinopathy of prematurity, which is when a kid is born premature. Um, and they put them in high oxygen to save their lungs. But unfortunately, that causes blindness. I think is what happened to Stevie Wonder, the
mm.
singer. So just series of diseases, mostly focused on the eye, but not exclusively. Yeah, that's what we.
Okay. I know I listened to one interview that Stevie Wonder gave once, and he had described it as like a big black spot on his eye or in his vision, and he could see things around the spot, and but that spot was like just it would get bigger and bigger
Yeah.
and bigger. Um, what I should mention one of our audience members actually here heard corneal transplants, and he said that sounds really sci-fi to him. So you're already in good with our audience here. So that's always good. We have some really good engagement sometimes. But uh, what is it like with the eye specifically? I mean, I heard you say that there's not that much blood flow in certain portions of the eye. So, if you could, how do they stay, for lack of a better term, living? Because, we always associate blood flow to living tissue.
So that's an excellent question. So there's two tissues in particular that don't get a constant blood flow. One
Hmm.
is the lens. The lens is in the middle of the eye and is responsible for focusing the light that goes through the front of your eye
Hmm.
and focus it into the back of your eye in a particular point that in human and primates is called the fovea. Um, uh, that the lens does not have any blood supply. Um, the lens is there, it's like an onion. It gets layers and layers as we get older,
Hmm.
especially when we're younger. So the center of the lens originates when you're an embryo. And then with time, more layers are forming and forming outside. For the lens to be translucent, it has to be transparent. It has to let the light go through.
Right.
Uh, so the lens actually has no, so most of the cells in the lens have no nuclei.
Hmm.
So the proteins that are there since the moment that cell got into place. So in some cases, 30 years, 40 years. Yeah. The metabolic rate of the lens is very small. Um, uh, and they
Oh,
do get
okay.
some they get some metabolites from the fluid they're swimming in.
Hmm.
The anterior part of the eye that bats the front of the eye is swimming in a fluid. It's called the aqueous humor.
Okay.
And the aqueous humor does have some nutrients and it provides glucose and other metabolites, but the metabolic rate is very low. So because those proteins are there, many from the moment you're born or later in life, they have to last a long time. That's why it's important that we wear sunglasses, right?
Yeah.
Because a cataract is nothing but the deterioration of some of those proteins. So a cataract is a disease. When people get cataract surgery, they're gonna have their lens replaced,
Hmm.
and that is because it has deteriorated. So UV light is bad for proteins. So although the metabolic rate is low, those proteins have been there they have to be there for as long as we live, hopefully.
Hmm.
So if you don't wear sunglasses, damage is gonna cause to those proteins, and damage accumulates. It might lead to a cataract. Yeah. So the other tissue that doesn't have a blood supply in healthy human beings is the cornea. The cornea is right in front of the eye. It's the first layer of tissue that the light is going to hit when it hits the eye. Of course, also has to be transparent. Yeah. The cornea gets the tears.
Mm.
So when you blink, tears are going to be covering your eye, and when you cry more, but even if you don't cry, you have tears. And the tears have nutrients, and those nutrients are going to are going to bath the front of the cornea, and those nutrients can be transported to the inside part of the cornea.
Mm-hmm.
And the same, the inner part of the cornea is bathing in the same fluid that the lens is, or the anterior part of the lens. That aqueous humor, and that aqueous humor provides nutrients for that part. So blood supply is effective, but not completely necessary for a tissue to survive if there are other fluids that provide
Mm-hmm.
the nutrients that those tissues require. Yeah.
I see. Um, so I know that you do uh, some specific research on a particular type of cells. I know them only by the name that I've learned them by, which is myanogs. Could you describe what those cells are? Because I know that they are a newly identified cell type.
Yeah. So myonog cells are a big part of our lab. It's not the only thing we work on, but we do work in them quite a bit.
Mm-hmm.
Myonog cells were discovered 30 years ago at Pickham by Mindy George Weinstein and Jackie Gerhardt.
Mm-hmm.
Um, Mindy George Weinstein and Jackie Gerhardt were looking for the cell that was the origin for the skeletal muscle at that
Mm-hmm.
time. And they looked at the myonog cells in the embryo. They were working with chick embryos with eggs, and they identified some cells, and they called them myonog cells because they found that they express two important factors. One is MyoD.
Mm-hmm.
MyoD is a specific skeletal muscle transcription factor, and then noggin. Um, and noggin is a protein that is very important for the development of the embryo, especially the central nervous system, the brain, the eyes, part of the central nervous system, the retina. So that was very important. So they named them myonog cells.
Mm-hmm.
And they found that they migrate to areas of stress. But Mindy George Weinstein and Jackie Gerhardt, at that time that we met, were at the Lankenau Institute for Medical Research, and I'm the Lankenau Institute for Medical Research is kind of a story had um, put together for an anniversary a cooking book, and they asked the staff members to sit down and sell the cooking book just because we had submitted some recipes,
Mm-hmm.
and they paired me with Jackie Gerhardt, who I'd never met before. And Jackie Gerhardt is an extremely intelligent person, and she always plans ahead. And she had looked me up, and they had seen the myonog cells in the retina and in the eye of the chick embryo, but they had never done any work with real diseases, the animal models of diseases. A little bit of work in the
Right.
in wound healing, but not much. My expertise was that I had a fair amount of experience on animal diseases and especially or models of human diseases, especially in the eye.
Mm-hmm.
So we started collaborating, and so well, let me look at them. And first thing we found was that the myonog cells were in the retina. So the retina is the part of the eye that detects the light, and is part of the central nervous system. It's formed mostly by neurons. It's all
Right.
the way on the back of the eye, and there is where the rods and cones are located. Those are the cells that are going to detect the light and send the signal back to the brain. So we found myonog cells in the retina. Um, we also found that that when there is a stress in the retina, more myonog cells, the number of myonog cells increases, and that they migrate to the areas when there is a stress. So not only the increase in number, but they migrate to particular areas of stress.
Mm-hmm.
So that was interesting. Yeah. So I thought, well, we know the numbers increase when there's stress. We know they migrate to areas of stress, but are they good or are they bad? Yeah. So we used a tool to get rid of the myonog cells and in a period of stress.
Mm-hmm.
And what we found is that when we get rid of myonog cells in a period of stress, the number of cells that die in the retina neurons that die increased significantly.
Huh.
So that was interesting. So we thought, well, they're probably good um, because when we remove them, there's more cell death. So the next step that we worked on was we purify them from a mouse and we inject them in another mouse.
Hmm.
during that period of stress, will they help? Um, and what we found is that the models of that animals were going to become blind, the injecting of myonuclear cells reduced the, the death of the photoreceptors by 40%.
Wow.
So we did that in a couple of models of blindness. And then I thought, well, if it works in the retina and the retina is formed by neurons, why don't we look in the brain? Yeah.
Hmm.
So we looked at an acute model of injury in the brain that is related to to a model of Alzheimer's disease. So I went to Australia. Jonathan Stone in Australia
Hmm.
had found something interesting, and that was that some cases of Alzheimer's or plaque formation related to dementia, he found that they were microhemorrhages. Um,
Hmm.
if you look at risk factors of people with Alzheimer's, many of them correlate to the risk factors or correlate to vascular problems. So the pulse is very strong,
Hmm.
and as you get old, your arteries don't dilate with the pulse as well, so can lead to microhemorrhages.
Hmm.
So they look at cadavers and they saw that where there was a lot of microhemorrhages, there were also plaque formation, and they designed this animal model to look at microhemorrhages in the brain, and they showed that 7 days after you induce microhemorrhage formation, you see plaque forming there.
Hmm.
So we used that model to inject myanox cells in the brain, and we found that 40%, almost the same number as in the brain as in the retina, 40% fewer neurons died when we injected myanox cells in those areas.
That's
So
fantastic.
yeah, yeah, yeah,
That's fantastic. I mean, you don't really hear about, those kinds of rescued cell numbers
yeah,
very often. A lot of times, uh, for the benefit of our audience, a lot of times, and especially in scientific journal articles, it's anywhere between somewhere around like 15, maybe 25 at the most I've seen, but
yeah,
almost normally it's around 8 or 9% of cells are actually rescued. 40% is fantastic.
yeah. That's great. That's great. That's great. We were very excited. Yeah, yeah, we're very excited. So yeah, that those results open the opportunity to some big grants. So for those members in the audience that are there, science is very expensive. Yeah,
Hmm.
science is very
Yeah.
expensive. You
Yeah,
you you need
that is one of the major facts that we tend to deal with.
some money is important to do experiments. So
Hmm.
we were lucky to get. So together we put an application, mostly to study myonuclear cells, not only in the brain but also in other models of cancer and things like that.
Hmm.
And we got a big grant. I think it was $2.3 million grant that we put together, and yeah, that has helped enormously. So we have maybe 22 students in the lab.
Wow.
You know that well because you're next to us, so you make a lot of noise. Yeah, they're
No, you
terrific.
guys don't make that much noise.
They're terrific, but yeah. So it's a lot of students there that are these are mostly medical students
Hmm.
that they volunteer the time.
Yeah.
They don't get paid for it. Maybe work study, which is a small amount of money,
Hmm.
just to volunteer the time to help the project, and they should take all the credit because it's their work. They are the ones doing the experiments late at night and
Yeah.
on the week. Yeah, and they work very hard. They deserve all the credit they get.
No, you're absolutely right. I mean, that's one of the best parts that I enjoy about the work that we get to do is, we get to share the scientific exercise with students that are not only interested but willing to engage in it with us. And I think that's a gift in all honesty, because you don't really see that a lot in certain labs. Most of the people that are there are only there to uh, get work done and then go home. And it's one of the reasons why I enjoy the university that we're in, and that's because there's a certain difference in culture than many of the other institutions around the area. And it's almost this, I don't want to say it's laid back because that's not necessarily the word I want to use, but it's more friendly. Um, it's more community based. We are. I don't want to make the joke of we're a family, but in a way, we are kind of a family, and I do enjoy that.
Yeah, I agree. I mean, look, I am a very lucky man. If I won the lottery tomorrow, enough money to retire, I still wouldn't
Yeah.
qualify. Yeah, I love my job. I think it's great. I truly mean it.
No, I
I will not.
completely agree with you. My wife says that if I stopped doing science, I wouldn't be a problem for her. I'd be a problem for myself because I wouldn't be able to live with myself. I'd have nothing to do. My hobbies can
Uh,
only take me so far.
I know. So that is wonderful, but definitely I'm with you. One of the things that I enjoy the most is interaction with the students. I mean, they're amazing. They just make our school is not easy, and they sacrifice so much of the time.
Yeah.
So much of the time, and it has brought me so much, and I learned so much from them. I learned
Hmm.
so much from them. They teach me every day. They're extraordinary people, and look, medicine these days is nothing but science, right?
Yeah.
No, that's just the truth.
No, you're
So
absolutely right. They are. I have this conversation with my students all the time. They are the practical appliers of everything that we study in a theoretical sense. They have to apply it, and I tell them a couple of times, listen, just because you apply it, you still have to understand it, and engaging in the scientific process, no matter how small, even if it's just something like record keeping, you gain a certain level of understanding of the process that goes into why we ask the questions that we ask.
yeah, I agree. I mean, look, I have a student now. They have the option on the fourth year to take 2 months of research.
Hmm.
This was a student that on his second year was ready to quit medical school. He was frustrated. He couldn't find his calling.
Mm.
He thought he had made a huge mistake, so he asked to take a break and come to the lab for a year. And he played a major role in our research. I mean, he ended up being one of the leaders of the lab. He will coordinate all the students. He was just for a while running a big part of the lab. So many
Hmm.
students, and um, and so it was nice to see him. I hadn't seen him for a couple of years, and he came back and said that that experience really brought him back to medicine, and he applies it every day. It made him trust himself managing people, and it's wonderful. Yeah, it's wonderful to hear. It's wonderful to hear. Yeah, yeah.
Yeah.
I think yeah, research is fun. It's fun. It should be
Yeah.
fun, and yeah, we're lucky. We're lucky. We get paid to do it, Joe, because you know,
That's true.
it's nice. It's nice to go to work with a smile on your face. It's
Yes,
nice to
no,
go to work with
you're
a smile
you're
on your face.
absolutely right. And we had discussed, with Dr. Balin a couple of weeks ago, scientific research for me has been one of the most rewarding processes that I've ever engaged in. I mean, don't get me wrong, the highs are high, but the lows, oh, they are there.
Yeah.
Um, but it it gives me so much joy to actually see a student gain that moment of clarity when they actually understand what's going on um in the work that they're doing. And it's that it I don't want to say it's a eureka moment because that's not necessarily the case, but just having that puzzle piece fit in for them so they finally understand the project that they're working on in a different light, in a light that we as the researchers see it already. That brings me almost no end of joy.
Well, you and I have the same approach. I think in some cases, the medical students that land in some of the labs, they
Hmm.
go there and they tell them, "Yeah, put this, pipette this blue
Yep.
liquid into this red tube, and that's it." But in my lab, the students own the projects.
Yeah.
They manage the projects from beginning to the end, meaning that they write. So when you are gonna create a project, sometimes you have to write
Yes.
to get approval for some of the experiments. They write it themselves. When we write papers, when we're ready to publish the scientific papers, the students write the first draft of the papers. They do the submission. I supervise the whole process, but they do it all. They design the experiment. They collect the data. It's a full immersion lab. And when I interview them before they come, I make sure they understand that this is not you're gonna enjoy it if you fully immerse yourself. If you just wanna do this just to tick a box, or if you're not
Yeah.
gonna embrace it, it's just not worth it. It's gonna take a lot of your time, and you're gonna be
Hmm.
miserable. So, try to understand every aspect of it, explain it, be able to explain to somebody else, understand every aspect. Because look, the best way to learn something is by having to explain it to somebody. So we try to get them to speak public and send them to as many conferences and do as many presentations as we can, so they're fully immersed in the project and it feels like they are their project.
I
Many
agree.
of
I
them
agree
come like,
100%. And I think that that's the probably the most pure scientific approach that I think that we as because we who work in the scientific field, we're also educators. We're educators of the people who come into our labs and work with us, and it's our duty, us those of us that are professionals in this space. It's our duty to make sure that we are not just doing the research, but also preparing the next generation to take up the mantle of what we're doing. And a lot of what I'm doing with a lot of the students here at our institution is I'm sowing that seed of, hey, you might not ever be doing research for the rest of your life, but there's a different perspective that you are going to gain simply by engaging in this process. Your brain will not look at a problem the same exact way. You're not going to ask questions the same exact way, and that's a great thing. You know, it, some students are very receptive to it. Like you said, some students they get into that space and they flourish. But I've seen some students that they really want to, and it just doesn't work. And my heart goes out to them. But unfortunately, it's kind of the nature of the beast in a way.
yeah. I try to warn them as much as possible
Hmm.
before. So I interview them before. I'm sure you guys do, and I try
Yeah,
to give them a heads up of what they're getting into. That is a full immersion experience. So
yeah,
I think they it takes 3 or 4 months to get a student fully trained,
yep,
where they are capable of using some of the equipment. Some of the equipment is very expensive too. Um, and if they stop doing research in 3 or 4 months, they wasted a lot of people's times, and that
right.
time going to be awkward to run into them when the system. So you know,
Yeah,
so
no.
I try to be to explain as much as I can during the first interview, so they understand that they're gonna get in something that it's gonna require a lot of time, and yeah, you can love it, but if you're not
Hmm.
sure, it's not something you need to do. Yeah, just for the sake of ticking a box. So
Right.
yeah,
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 no, I completely agree with you when it comes to that. The ticking the box idea. Unfortunately, the students that we have, because their time is taken up so much. A lot of times they only go into the research just so that they can add that onto their resume. Um, and some of them they initially go into that. I don't know if you've experienced this, but some of them come in initially for that, but then they find that moment where it kind of clicks for them, and they suddenly an old professor of mine used to call it getting bit by the research bug. You get that moment where it's like, well, now I want to ask another question. It's like, well, we can do that. We can absolutely ask another question. That's kind of the whole point here. Ask more questions. And
that is wonderful.
your smile says it all. That's exactly the kind of thing that we love to see. And I stress to my audience here, or our audience in this situation, um that's kind of always what we want, is we want to make it so that people understand that we are just people, and that we really enjoy what we do. We have a excitement for it.
Yeah, I mean, look, yeah, there's nothing that the scientists love to talk more about than science. Yeah.
Exactly. Exactly. No,
So
you're absolutely right. So I
so
already
I don't
have
know.
a
Yeah, I know.
question
I
for
know
you.
the other professions. I don't know how.
Yeah.
I'm sure the jobs that are wonderful around there, and but yeah, most scientists I know they love they love they love what they do. But look, it hasn't been a road without problems.
Right.
The stories that they can tell you. So when I was so I'll tell you. So not everything is, yeah. For those in the audience, and I'll tell you, yeah, that, unlucky, we love it, and and truly Pickup is a great place to do sciences. It's a lot of fantastic people like Joe, and I'm really good friends.
Thank
But
you.
not all places are like that. I was a postdoc at Harvard in a lab,
Oh boy.
and it was a rough environment. I had a sleeping bag in the lab, but look, I was excited to work there, and I think
Mm-hmm.
most people were. But the competitiveness of some level
Yeah.
was really difficult. So when I arrived, a couple of 2 or 3 of us arrived at the same time, and there was a postdoc that had been there for many years, and she had made some striking discoveries. But she got, she was the star of the show, and we came in hitting hard.
Mm-hmm.
And we wanted to do findings, and we worked hard at it. And so somebody's experiment, she had these animals in oxygen, and somebody was coming at night and turning off the
Oh
oxygen
no.
cage. And we couldn't figure out who it was. And we all had suspicions. And the PI, the principal investigator who was
Mm-hmm.
running the lab, bought one of those powders that you see in CSI that you only see what you believe.
Yeah.
Um, and she put it on the gauge, on the oxygen gauge. And the person that came to turn off the oxygen did that, and then went to her computer to check her email. And so then the next day we found who it was, because of course it was her computer, and she admitted it. And and she had no reason other than she just didn't want somebody else to succeed,
Yeah.
somebody else's to succeed. But the funny thing is, the PI could not kick her out of the lab
Yep.
because she was about to get a publication in a big impact journal, and she needed that for her own career. So
Mm-hmm.
then they had compromising, and she could only be there from 9 to 5 when there was people there, and she had limited. Yeah, so she had to work supervised. I know.
Oh man. So
So then,
to shed some light on this for our audience, postdocs, you're already a PhD, and that's kind of your time to build the foundation of your career. Usually you're just kind of given carte blanche to do almost anything that you need to do to get the job done. I it's so funny to me to hear that a postdoc had to be limited on their time. I've never heard that before.
yeah, I mean, it was extreme. I mean, she was sabotaging somebody else's experiment.
Jeez.
She was also coming and using. We were growing some cells in plates. So you grow
Mm,
cells in plates and you put them in an oven,
yeah.
keep them at the right temperature so they grow right. She would come in at night and pouring ethanol on them so that they would. I mean, she was something special. But guys, when you put people under a lot of pressure,
Yep.
they do things that normally they wouldn't do. People do
Exactly.
war. People in extreme circumstances will behave in ways that they shouldn't be doing. So, not everything is wine and roses. Yeah.
Yeah.
Overall, but that's like anything in life. But overall, it's quite a good job when you land in a good place.
Hmm.
Yeah, science is it can be terrific. But
Yeah.
yeah, so some environments are push people to
No, you're
behave
right.
in ways that they shouldn't. Yeah, they shouldn't.
So we already have a couple of questions for you that have popped up. Um, Number one, how often do you get asked about the carrot myth when it comes to carrots benefiting your eyesight?
Uh, so look, I don't get asked that question that often. Look, I love that you guys love talking to science, but I tell you, I am not the highlight of the party. I am not. First, I have a very thick accent, so usually when I start talking, people go, "Oh, you're gonna get to drink," and then if I talk science, yeah, I'm gonna end up in a corner, yeah. So uh, you know, carrots do have a vitamin that is very important for is part of the process in which we capture light. Um,
Hmm.
so vitamins, most vitamins, vitamin D is not, but most vitamins, you can only get them through nutrients, and that's what
Yeah.
they call vitamins, right? Um, so if you don't have that nutrient, if you're deficient on vitamin A, you're gonna have a problem with your vision. Uh, but yeah, so if you're deficient, then carrots will help you. if you're not deficient in it, they are not gonna help. Yeah, they're not gonna help. You adding something
Right, it's
something
only
you
going to
already
bring you
you're
up to the base level
yeah
that you need.
yeah they're not gonna help. What things that you can do? I can tell you that when you read something, or when you're on your cell phone, or you're on a computer, um, your lens is under stress, and is
Hmm.
under stress, physical stress, meaning that that lens is gonna have to accommodate. The lens is not always the same shape, depending of the tension. It's got some ligaments that extend it or relax it, and those ligaments are attached to a muscle.
Hmm.
So the reason why you get a little bit tired, your eyesight gets a little bit tired when you're looking at a computer or a cell phone, is because that muscle, for you to focus on that close vision, that muscle is constantly contracting. So you're flexing that little muscle to help the lens to be in that shape. Um, and that's why that muscle gets tired too. That's why they recommend that you focus on something after a few minutes of working on a computer. You focus on something that's far away. Because focusing on something far away will make that muscle relax for a little
Hmm.
while, and that is always good. So those are good things. You can take care of your vision, that you can be proactive on.
Hmm.
I can tell you that sunglasses are good. Sunglasses are good.
Hmm.
Sunglasses are good for many things, but not only for the lens, as I explained earlier, but also because they reduce the number of UV light, like the amount of UV light that hits your retina, and that's always a good thing.
Hmm.
Um, so sunglasses help protect your vision. So there are things that you can do practically that will help you with that. Um, Look, there are reports of antioxidants helping in some eye diseases. And that makes sense because the retina has the highest metabolic rate in the body.
Hmm.
It uses more oxygen metabolites than any other tissue in the body. Um, and Joe, you were asking me this before the audience came. I'm
Yep.
talking about the reactive oxidative species, and one of my students asked me about that. So you do produce a lot of reactive oxidative species in the retina,
Hmm.
and some sort of antioxidants, in some cases, will help with some retinal degenerative diseases. I know, because I've seen the studies and I've been part of them, that saffron uh, is very effective to reduce
Really?
some retinal degenerations. Very effective.
Really? Saffron?
Very effective. Will help with retinal degenerations,
Oh, that's awesome.
macula degeneration, retinitis pigmentosa. Saffron works really well.
I can't wait to
We're
tell
doing.
my wife we'll be eating more paella.
You need more paella, you can come to my house and give it to you. In this
Oh,
case,
lovely.
it's a high amount of saffron. It's more than you will have in a paella. It's
Hmm.
a, it's a pill with a fair amount of saffron in it,
Oh, okay.
around 10 milligrams per kg. Um,
Oh.
so that's how much they take. And it's sold commercially in Italy. And we have a student arrived from Italy, just
Hmm.
arrived to my lab. You might have met her, Federica,
Yes.
who's going to do a study. We have a model of glaucoma. Is going to do a study on saffron and glaucoma. So,
Oh, that's
so
fantastic.
yeah. So, and I've seen the study. So it's not like I've seen it. I've seen it work with
Hmm.
retinal degenerative eyes. This clinical studies working on it. So is and this commercial companies that will produce it. So that really works. How it works, we aren't sure. It seems
Hmm.
to prevent inflammation. Inflammation is part of almost any
Yeah.
complication in the body. Um, so maybe that, but we're unsure how it works. We're trying to study the mechanism, but yeah. So there are things that help. There are things that
Hmm.
help. Carrots, yeah. I mean, you have some antioxidants in there.
Yeah.
They surely help more than probably a steak. Um, but yeah, in terms of vitamin A, you reach a threshold, well, after
Yeah.
which you don't get any more help
I believe
from it.
vitamin A is that water soluble or is that fat soluble? I forget.
It's water soluble. Yeah, I
Okay.
think it is. Uh, so
Um, so one of our audience is actually from Australia, and he was telling me, he says he mentioned that you had a colleague in Australia, and he wondered if it was his brother's boss who manages the brain banks at the Department of Neuropathology at RPA Hospital.
so I did my PhD in Australia.
Hmm.
So I was born in Spain, and then I'm a chemist, a biochemist, and then I moved to New Zealand. I worked in New Zealand for a while
Oh wow.
in diseases that affect oranges and apples. So it was very different to what I'm doing now. And from there, I moved to Australia. I was working with Jonathan Stone. Jonathan Stone is a member of the Academy of Sciences of Australia,
Hmm.
um, quite a character. And Jonathan Stone did a lot of the initial work. First, he discovered some cells in the retina too, some ganglion subtypes of ganglion cells. Those
Hmm.
are the cells that the axons will form the optic nerve. The optic
Right.
nerve is at the back of the eye. So when you see a scary movie, when somebody's holding an eyeball, they're holding it from the optic nerve. That optic nerve comes from the nerves of those cells, the ganglion cells. They all get together. The last cells that are responsible for sending the message from the retina to the brain.
Hmm.
So Jonathan Stone discovered the cells. He did some of the initial studies in saffron that I'm talking about. He discovered that during the development of the retina, there's a period of stress called the critical period, in which a lot of the cells, the neurons are going to die. So we're born, that happens in the brain too. We're
Hmm.
born with more neurons than we need.
Right.
Um, and some of them will die after birth. In cases of diseases, degenerative diseases, that moment of cell death never stops. So it
Right.
keeps going up. And that's how you get people with retinal degenerations. But in many cases, it's just a peak that goes down once you find that balance between what you need and what you have. Um, so I was with Jonathan Stone at the University of Sydney. And then from there, Jonathan moved to the Australian National University in Canberra.
Mm.
And so the last year of my PhD was in Canberra, and I actually graduated from the Australian National University in Canberra.
Wow.
So there's very good research done in Australia, very good
Hmm.
research in the eye. I have to say they're really good. Some of this research is done, uh, but there's a fair number of people that do research in Australia,
Hmm.
and I don't know if I'll know the name of your brother, but yeah, we didn't at that time. We didn't do a lot of work with cadavers or human tissues. So probably, and this was 25 years ago, so it's probably unlikely. It's probably unlikely, yeah, that I met him, but yeah, it's good to hear from you. Yep. Yeah. Yeah.
So we have another question for you. So people who can discern color on a deeper level, can they discern different shades more so than others, or is that a physical difference in their eye, or more just a personality thing? So I guess it's a perception of the color that the people have.
So yeah, so um, you have in your retina photoreceptors. The photoreceptors are divided in the cones and the rods. The rods,
Hmm.
as I said earlier, are responsible for night vision, vision under the stars. If you go outside now, unless you're in Australia, you probably have the stars under you, but you can still move around. So those are the rods. The cones detect light in a particular wavelength. So you have cones that detect light in the red spectrum, some in green, some in blue. Uh, some people are able to detect, and we think it's because they probably have a mutation in one of the proteins that allows them to capture a little bit more of a spectrum than somebody else. And Jackie Gerhardt, uh,
Hmm.
Joe, she can detect some I think far red. She can see
Yeah.
far red with her light, with her eyesight, and I cannot do that. So how does that occur? Um, it's an interesting question, and I don't know the answer of that. Um, but
No, that's.
probably she might have. Yeah, so the protein that detects the light. So the light hits a protein, and that protein changes
Hmm.
conformation. So she might have a small mutation in that protein that makes it more sensible to certain wavelength. But this is just a hypothesis. I'm speculating here. I'm not completely sure how she has it. The opposite is also occurs. So some people are colorblind.
Hmm.
So some people are only have two kinds of cones, or less or fewer of the other cones, and they all gonna miss certain certain wavelengths, so they might not see the red or they might not see the green. And that's what color blindness is. But there are some people that are able to see more of a spectrum than the rest. And of course, animals can, like animals can see
Yeah.
UV light and things like that. So, but humans uh, have better vision than most animals, better vision
Hmm.
than most animals, and especially the ability to focus is quite good. And it might be because we don't have very strong jaws or teeth,
Mm.
or we are not very fast. Because we used to jump from branch to branch, so you don't want to miss the branch. Um, so yeah, is quite interesting, that we have quite good vision. And yeah,
Hmm.
yeah,
No, I mean, so one thing that I started this whole series on was to kind of show basically that we scientists are just normal human beings, and we're not the supervillains that you find in movies and TV show. Um, so could you tell us real quick, what got you into science in general? Why science? Why not music or art or history or math or any other topic of study or
Joe, if you
practice?
heard me sing, yeah, you'll understand. Mus- Music is not my calling. It's not my calling. um, I have to say, I've always loved science. My father was a clinician,
Hmm.
and I got a chemistry set. I'm a chemist originally, and then a biochemist, and now I do mostly neuroscience. But um, I got a chemistry set that probably wouldn't be allowed to have these days. Uh, and I found it fascinating. I thought it was a lot of fun. I always had an interest in nature. And yeah, it was something that's I always found it interesting. Science was always something I really found, and the more I knew about it, the more I liked it.
Mm.
So really, I always liked it, but I fell in love with time. I have to say, I fell in love with time. I was lucky to land in some critical moments of my life in, in the lab of people that really helped me, and they were extraordinary scientists. And Jonathan Stone is one of them.
Hmm.
Um, I was volunteering in his lab, um, and at that time I was washing dishes. I was an illegal immigrant in Australia for a year, and volunteering in his lab. And after a year, he asked me to join his lab and do my PhD over there. And I thought what he was doing was groundbreaking, and it was. So I was very lucky to land with really extraordinary people that helped me by to nurture that love that I had with science. The only thing that maybe could have challenged my scientific career was probably rugby. I play a lot of rugby for the Spanish national team, which is not very difficult. Unlike Australia, you don't have that many people playing over there. So that that something that probably for a while took me away from science, but
Mm.
always came back to science. Yeah, it's like a comfortable bed. Yeah, it's a
Yeah.
good place to spend time, and yeah, it's a good place to stay. But we get paid
I
to think, and
yeah.
that is a luxury. Yeah, that's a luxury. So
Yeah, that's definitely something that I've always enjoyed about it. I've always likened it because my brother's a Catholic priest, so I've always likened my experience in science to a vocation. It's always been that thing that kind of just like grabbed at me and kind of pulled me in its direction.
I bet those you guys have good, interesting conversations over dinner. Yeah.
Everyone assumes that we have these angry
No, I don't think so.
conversations that we scream at the top of our lungs about something or other. Whenever we talk about science, he kind of is quiet and lets me talk, and whenever he talks about theology, I am quiet and let him talk because we both my father, who was a history teacher, was very good at debate, and he would always say, "Listen, you don't go up against somebody when it's their wheelhouse, when they are setting the battlefield again, because he was a history teacher, so it was a lot of war references. Uh, Listen to the person who knows better was always his philosophy. You could always listen to the person who knows better a little bit more, and you'll learn something better for that.
Yeah, that's a good piece of advice, and that's a
Hmm.
good approach. I think that's yeah. If everybody debated like you guys do, we'll probably be better off. I don't see. Yeah, I'm not a religious person, but I don't see incompatibility between religion and
Hmm.
science. I'm not people who were religious and they had their faith, and then they have the science, and they were able to compartmentalize those things. And I think they were extraordinary scientists, and they were people. So whatever they were doing, it was working for them, and I'm glad it was. Um, so yeah, yeah. But a good debate is always a pleasure. And it's yeah, yeah.
Well, I mean,
It's good to
that's
listen.
kind of
It's
the
good to listen. Yeah.
best. It's kind of the best litmus test for your theories. If your theory can't stand up to a debate, even the simplest debate, it's not going to hold up in any peer review or scientific journal whatsoever. So I'm always up for discussing a theory that I currently have with somebody, because they're, well, number one, they're going to give me a perspective that I haven't thought about. Because like you said, you were a chemist and then a biochemist. I am terrible with biochemistry. If you start asking me questions about biochemistry, I curl up in a ball to kind of protect myself. But there's a perspective of the problem that I'm working on that you, as a biochemist, may actually have an answer to that I haven't even considered yet, which for
That's
me is
true.
very important.
I haven't done much biochemistry for a while, but
Mm,
I can tell you, since we're talking on some of the aspects of science that are not as good, somebody said that science advances every time a big scientist retires. Yeah, they're holding the ground. I can tell you that some of the discoveries that we have done in the lab have raised a lot of eyebrows,
mm,
and that we have faced some skepticism. And I wish people like you welcome a new idea and you
mm,
challenge that idea and you accept the facts when that idea stands the
yep,
ground, right? Um, but that is very unusual. Some people that have been building their careers
yeah,
over something that might have not been, a full truth. In some cases, um, it's not completely wrong, but it might be, part of it might not
mm,
be completely right. So for us, that has been challenging. In the case of myonox cells, we showed that myonox cells are very important for a complication that comes after cataract surgery. So
mm,
when you have cataracts, they remove them, but they leave a capsule. So the lens is involved in a capsule. And usually
right,
what they do, they leave the capsule and they remove what is inside and they put a new one.
mm,
Now, in many cases, you have a secondary cataract. It's called posterior capsule opacification, and you need surgery or laser to remove it.
mm,
We showed very strongly that was caused by myonox cells because myonox cells help with vision. But one of the problems that we have is the turn into myofibroblasts. Myofibroblasts are muscles that express contractile proteins.
mm,
And the fibrosis. So they can lead to fibrosis. So the story is not all that good. It's great that they're neuroprotective, and you have 40% more neurons when you take them there.
yep,
But it's also not good that they can lead to fibrosis.
mm,
So that's the aspect that you have to know about myonoid cells. They're not a miracle, like many things on science. Um, so we showed it. But God, the people working on the lens were so against it. It was so some were extraordinary and they accepted facts and we established great collaborations. But there's a lot of people that were really really difficult to deal with. And turn their back to evidence.
yep, yeah. There's pushback.
But yeah, so and I'm okay with discussion. I think that it's very healthy and checking somebody else's results and reproduce them. All of that's wonderful. But you know, this some science has advanced to a lot of people that made mistakes and then
Hmm.
were corrected and that's how we move on. It's a thing to celebrate.
Yeah.
But yeah, it can be tough at times. Not everybody has the approach that you have, and you know, again, people are need money to
Mm,
do science. So if somebody discovers something that's going to challenge their
yeah.
belief or their work for 20 years, and it's going to be difficult for them to make money and keep that lab open, and that means people that work for them,
Hmm.
um, they can be they can be yeah, they can be a little bit stressed about the situation, and
Yeah.
we all know what the stress leads to people do things that they wouldn't normally do.
True. True.
You know, but
So could you, because you mentioned it earlier, could you describe your experience with being an immigrant? I'm assuming that your home country is Spain. I'm assuming. Um, could you describe your journey as an immigrant for us?
I went to New Zealand when I was in Spain. I finished biochemistry, and then I was playing out of rugby then. So at that time, uh, I had just graduated with my master's in biochemistry, and I wanted to play rugby in New Zealand. It's a good country for rugby. So I went there, and I found a job in a lab, and
Hmm.
and yeah, I I landed in New Zealand, and I worked there for a year as a technician in a lab. It was a good year. I couldn't speak in English, and in New Zealand you don't have a lot of Spanish speakers.
Hmm.
So although New Zealanders are very good people, they are very Anglo, and in terms that they are, you don't see a lot of people holding hands. It's a country that is beautiful and the people are very nice, but coming from Spain, the cultural differences were big.
Mm,
But you always find your crowd,
yeah.
and they were welcoming. But it's difficult to communicate. It takes around 6 months when you land in a new country, if you don't speak the language, in my opinion, to reach to the point where you can understand a joke.
Hmm.
So the first 6 months were rough, um, but it was a good country. It treated me well. From there, I moved to Australia. Um, Initially, I was going to be working, volunteering for 3 months, as how long my visa was, in a lab, doing studies on biochemistry on the corals, the connection, the symbiotic interaction between corals and algae.
Oh,
Um,
wow.
Corals live in symbiosis. Some corals live
Hmm.
in symbiosis with algae, and they exchange metabolites. Um, that didn't work out that well. Somebody was making my life really hard in the lab, and some people that saw me working hard recommended me to Jonathan Stone's lab. Arrived to Jonathan Stone, and he asked me what I wanted to do. I said, I just want to learn. You don't need
Mm,
to pay me. I'm just gonna volunteer here. So I was going there to join. By then, my visa had expired. So I was volunteering at Jonathan Stone's lab from 8 to 5, and then from 6 to 1 washing dishes
mm,
in a restaurant in Sydney. Um, you know, it was fine. I didn't have a family. Most of the time, um, I ate at the Hare Krishna. They
mm,
provided free food. They had a soup, like a free food kind of service
mm,
for homeless people and people that were poor. So, but it was good food. And I lived in a house with 12 roommates and one bathroom. So it was an experience. And then after a year, Jonathan Stone Lab, he asked me if I wanted to do a PhD with him, and I didn't have money to pay. It's very expensive to study in Australia.
mm.
And so as an international student, I didn't have the money. So he gave me a scholarship to study there, and he gave me enough
Wow.
money to not have to work in a restaurant anymore. So I was more expensive to him than a postdoc. He was very generous. And Jonathan and I still collaborate. I go there often to Australia. We've done some really good studies together. He's a good friend. We organized a conference together 6 months ago in Sydney. And when I was about to graduate from my PhD, somebody from Harvard asked Jonathan if he knew somebody who could work in the retina. He recommended me, and yeah, I went to Harvard, and I was in Boston for a while. It was a new country, so every time you move, it's always there's a period of adjustment.
Hmm.
I love the United States more than any other place I've ever lived in. Um, although I have to say, people were really nice to me in Australia and really nice in New Zealand. But I connect more with Americans. I think Americans are more.
Mhm.
Look, Australians and New Zealanders are very Anglo for a Spaniard. Okay, yeah. I lived in New Zealand. I don't remember many people shaking hands or kissing on the street or things like that. So, um, Americans are more casual, and it
Yeah.
was I don't know. For some reason, it was easier for me to connect with people in the United States. No place is perfect. Um,
Mhm.
so if you wanna focus on the negative, you're gonna focus on the negative
Yeah.
anywhere you live. But for me, the United States has been a good place. And there's always problems. People. You choose the life of being an immigrant, and there's always gonna be problems. You're always the person with the accent. You're always gonna be, you know, you're always gonna be, the person that doesn't understand some of the things. So my daughter, I have a 2-year-old daughter, and I've never gone to school in the United States. And when I ask people, and people are telling me a story, they keep referring to grades instead of ages. And
Yeah.
I don't understand what a grade. A grade is not an age. Yeah, sometimes I'm really confused. It's a grade 5, so they were 5. No, they were not 5. They were 10. Some were 9, and say, "Oh God!" But so
Yeah,
that took me
we
a
we
while.
do things a little weird here in the U.S. I do apologize that we kind of have
Nah,
our
nah,
own
you don't
way
have to
of doing
apologize.
things.
I'm the one that should be learning it. You like the system. So look, there's always experiences that you go through that teach you.
Hmm.
So for instance, when I first moved to New Zealand, they invited me to a party. Yeah. And in Spain, when you organize a party, you provide food and drinks. So I was
Yeah.
used to that. You don't ask people to come and provide and bring food, like a potluck style.
Hmm.
So this was what they called it at the time. Say they sent an email and said, "Bring your own dish." Um, so I looked dish in the dictionary, and I saw plate. So I thought, these people are having so many people at the party, they don't have enough empty plates. So I brought an empty plate to the party. Yeah. So
Pardon,
yeah,
pardon my
but they
laugh.
were.
I'm sorry.
Yeah, I know, I know. But they were good people, and they took it with good humor, and I think as I said, being an immigrant, it could be an advantage sometimes, and it could be a
Mhm.
disadvantage, and it's your own choice how you're gonna focus on what you're gonna focus on, the good or the bad, like with anything. So I try to focus on the good. Yeah, yeah, but
Good.
yeah.
I'm glad that you were. I'm glad that you had at least a, because I know that there's always good and bad in every situation. I'm glad that you at least had a good experience from what it sounds like.
Yeah, I mean, look, like every experience in life, you're gonna
Mm.
have things that are gonna annoy you, but that could happen
Yeah.
to me in Spain too. It's so, yeah. So you just have to find your own crowd.
True.
You learn a lot from living in other places. I mean, it opens your
Hmm.
mind. You learn different ways of living, different perspectives on life, and it's overall a good experience. That's one of my favorite things about science. Science really promotes people traveling to other places and
Yeah.
spending time in other labs. If you're a lawyer, it can be difficult to work as a lawyer somewhere else. You might have to pass the board, the
Mm.
the bar exam. Same if you're a clinician. Well, science, you don't, right? You say you're a
Yeah.
biochemist, and basically people say, okay, come over here
Hmm.
and do whatever you want. And that is something I really love about science. They
Yeah.
really encourage people get into other places, learn new techniques, travel. Um, is wonderful. It's wonderful. Yeah, yeah. It's one of my favorite things. Yeah, about it. So
No, I completely agree. Could you, for both me and because this was a question from our audience, how is science different, at least either in community or in the way that it's actually conducted, between different nations?
that's a really good question. That's a really good question. Spain has a public education system that I think is exceptional. Uh,
Mm.
it has really good universities that you come out with a debt, but it does not have a follow through. So
Ah.
after that, you're on your own. It doesn't invest enough on the scientists. So they produce the scientists that go anywhere or other countries.
Hmm.
So it's little investment on science, and not at the level that you have here. Um, Australia and New Zealand, most of the focus, I was working in agricultural research at that time, so most of the focus was really in terms of production, avoid diseases. New Zealand, one of the biggest markets for New Zealand crops was Japan. And
Yeah.
Japanese pay a lot of money for the fruit, but they want it perfect. So
Yes.
you couldn't have spots or anything on the fruit. So that's what we were doing there. So it was really focused on the commercial aspect of what you were producing in the field I worked.
Hmm.
Australia was a good place to do research in this, but it does not have the resources or the biotech industry that you have in the United States.
Right.
It's mostly, or at that time, was mostly academic research. Um, I I thought Australians had very good universities. They had developed very novel um, approaches to some problems that other people hadn't thought of. I think Australians are very good problem solvers, and Australians are travelers. So that was an advantage. A lot of the people who work in Australia had traveled to other countries and gotten
Hmm.
other ideas and brought them back to their own country. So that was very good. It's quite normal for Australians to live abroad for a few years. The United States just has a lot of money. It has
Yeah.
a lot of money, uh, and it's a very entrepreneurial country. Here, people really is very easy. I've started a couple of, you know, biopharmaceutical companies. Some of them are still there, and I worked for some for a while. It's a very easy place to transition from a discovery to having a company to move it forward. There's lots
Hmm.
of help, and that's really wonderful because you know some people can get frustrated in Australia and Spain when they have good ideas but they can't carry them forward. It's difficult to get the money to move them forward. It's
Hmm.
difficult to do the studies. When you find a discovery and you want to move it into a clinic and treat patients, that costs an enormous amount of money. Um, so when people talk about big pharma and how they can sell things, you have to understand that it takes a lot of money, a lot of time, and a lot of failed projects to get
Yeah.
to that point, right? So I'm not justifying, but just there's something that you have to consider. Um, that in the United States is always available, or available. If you have something really good, it's relatively easy to transition. That is
Hmm.
nearly impossible in Spain and really difficult in Australia. So that translational science is definitely very good in the United States. And um, the universities here have quite a bit of money, so
Hmm.
you put on money into research. So that is a real advantage.
Yeah.
In terms of disadvantage of the United States, I don't know. You know, in Boston wasn't as much. Boston is very cosmopolitan, but I find the scientific community in Pennsylvania has not had a lot of very global experiences.
Hmm.
A lot of people have studied in Pennsylvania. They did the postdocs around here or close by, and they work in academia within
Yeah.
Pennsylvania. And that I think is probably not a good thing. So, for instance, I just brought yeah, I brought three scientists to Pecom, right? And one of
Hmm.
them is a PhD student. She wanted to stay here for 6 months. Pecom does not is not has never requested the capacity to give a visa to somebody
Yeah.
for 6 months that is coming to do a scientific collaboration. For me, that is, I mean, the Italians didn't understand that because they're always bringing people from outside.
Right.
The
Yeah.
Spaniards will always be doing it. So that was for me mind blowing. I mean, I just could not understand how somebody didn't set up it's a university and you have research. Why wouldn't you have applied to be able to sponsor researchers to come for 6 months?
Yeah. Three
Um,
months is a very, very short time for
well, she
scientific
comes on a
research.
tourist visa.
Yeah.
So now she's going to have to go back. So she was stopped at immigration. We
Hmm.
had a big problem. Even though she's a volunteer, I don't pay her anything, but she, you know, then she's gonna have to come back in September, and that's gonna cost her money. She's a PhD
Yep.
student. She doesn't make that much money. So, so that, um, yeah, I think Pennsylvania and Philadelphia is a great place to do science. There's a lot of industry, biotech industry
Hmm.
here, but it does lack a little bit of or at least in my experience, in some institutions, Lankenau, the Institute of Medical Research, and Pickham, there's a lot of, oof, other than the guys that come, the foreigners, there's a lot of, yeah, even those have studied here, and they got in here through, being to UPenn
Mm,
or one of the local institutions, so that is, look, talking to people that is different than you is always enriching, right?
yeah.
And going somewhere else is always enriching. So I think, yeah, I think, yeah, I'll say probably one of the few things that I don't like about science in the United States or in well, in some places, as I say, Boston is very cosmopolitan. So Boston, you
No,
have a lot of
I can completely understand that.
affiliate.
You know, I that's one of the things that I have enjoyed about science, at least in the United States, is that I've been able to meet several people from different countries. I got to enjoy time with someone who was French. I got to enjoy time with someone who was Japanese, someone who was actually from a little village in Siberia that when he finally told me how the level that he had to get back to his village, he said he had to take a commercial plane, then he had to charter a plane, then he had to take a sled back to his village just to get home. And he said, you know, it is always cold, it is always freezing. And I said, well, how do you live? And he said, we live heartily. You know, I'm very grateful for all of the different people that I've gotten to meet over the years. And I've even gotten to meet some people from Germany as well. And one thing that I've noticed is that you know science, kind of like how the Catholic Church used to be, we kind of have a universal language in a way,
That's true.
and I really do enjoy that fact because I can, even though I might not speak the language perfectly, uh, I can still have a conversation with someone about science simply by pointing at pictures and maybe drawing something. And to a certain extent, you're going to understand what I'm talking about.
That's true. That's true. So if I were to recommend somebody who is in high school, who has the itch to travel, right,
Mm,
and wants to live in other countries, some people go into teaching English, and that is perfectly fine.
mm,
But it's hard to make a career the rest of your life teaching English. There's very
yeah,
limited options, very limited growth options. I would say science is a good place.
yeah,
Um, because nobody's going to ask you to take an extra exam to be a scientist in France or in Germany or
yeah,
Russia or in Japan. You just come with your degree and you can work.
mm,
Um, and that is very unusual for many professions. Very unusual. Many of them require some accreditation, and you have to take local exams to practice your profession. So if you have that itch, it's definitely an advantage. That I think is a good thing. If you have that itch,
mm.
you don't travel, you still can do science in the United States, and it's great science place. So yeah, yeah.
So, for you, how has new technology impacted your research? Because obviously, the technology that we have today is not the technology that was there when you were a bright-eyed, uh, idealistic scientist walking in on your very first day.
So for us, has dramatically changed the way we approach it, and changed it for good. So for instance, a lot of our work, look, tomatoes don't have eyes. So when
Mm,
you work with eyes and neurons, you have to work with animals that have eyes, right?
yeah.
So a lot of our work has a mice. Uh, I don't particularly enjoy working with mice. I could set them free in the field, but if you want to cure vision, you need to work with mice because they have eyes. um, so for us, technology has allowed us. So we use a machine called OCT, which is optical coherence
Mm,
tomography, and that is just to make it just very basic. Would be a mix between a microscope and an ultrasound. Um, so with this allows us to see um, the retina and how an animal is getting blind with time,
mm,
the same animal through time, right? So in the old times, before we had this technology, we would have to sacrifice the mouse, look
mm,
at the eye, take the eye, look at the, and in microscopy, look at the eye, and you have to sacrifice, you have 5 time points. kill them
Right.
when they are 4 weeks, when they are 5 weeks, 6 weeks, 7, 8 weeks. You have to sacrifice an animal every time. But this technology allows us to follow that same mouse through time and see how it's becoming blind,
Hmm.
and take images, live images of the retina. It has allows us to determine how well. I mean, one of the problems is that you cannot put a mouse in front of a chart with letters and numbers of different sizes, right? You can't.
Well, you can try. I'm
You
not
can
saying that
try.
you'll
We're
get a
gonna
really
look at
good
you like, what are
result,
you
but you could
saying, right?
try it.
How do you know how well an animal can see?
Right.
So one of the ways that we do it is a functional study. Is called electroretinogram, which is like an electrocardiogram but for the retina. Uh,
Oh, that
and
sounds
that we put the
amazing.
animal, and we put a series of probes, electrodes, and we put a flash of light. And that flash of light is going to initiate a signal,
Hmm.
and we can measure the intensity of that signal, and that will tell us how well the animal, how functional that retina is or not.
Mm.
So for us, technology has been, it has completely changed the field in the last 20 years. It's really, and for good, we don't need to use as many animals, which is something
Hmm.
that I like. So I think every animal's life is valuable.
Yeah.
And just the we being able to, there's difference between animals. So if you can follow one animal all through, instead of having to use five different animals, you reduce the experimental error from one to another. This is you're very good at statistics, and you have helped us a lot. So, you
Right.
know, every advantage for us. Technology has really, it's costly, but it really has really, changed completely the way we do research. I mean, now we used to have to do PCRs ourselves to determine the genotyping of a mouse,
Hmm.
so that whether a mouse has a gene or not. That's you do that by genotyping. We used to have to run it ourselves, and that was a whole 2 days of research just to look at them. And now we can
Yep.
send them for $20. Somebody does it in California, and we
I
get the
I
results the next day.
I know what you're talking about. I used to do DD Sanger sequencing, and that was I remember watching on TV with my mom one day, and they were. I've said this before on stream, so I apologize to my audience because they've heard it like 100 times, but I remember, I was watching some crime drama or something with my mother, and they gave like genetic material to somebody, and then maybe 20 minutes or 5 minutes, 5 seconds later, they come back. We got a result. We got a match, and I'm like, what technology are you using that
Yeah,
sequenced the entire genome that fast? You'd be lucky if you got a sample next week.
that is right. That is right. Science and love are very different in life than in TV.
It's very true. It's very
In this
true.
day.
So, one thing I did want to ask you, because I've been seeing a lot of, and obviously COVID has impacted us in multiple ways, but I've been seeing a couple of different things, especially with long COVID, about its effects on things. How has COVID changed your research and researchers that are similar to you? So basically, I guess what I'm asking is, are you now looking into potential questions in COVID as well as what you were currently doing?
We're not.
Okay.
Uh, we are not incorporating COVID studies into our research plan, and simply because, quite frankly, I have more results that I can publish. I have time to publish.
That doesn't
Um,
sound like a bad thing, to be completely
it's not a bad
honest.
thing, but I am the bottleneck right now. I am the
Yeah.
bottleneck, and the students rely on those publications to
Hmm.
apply for residency programs, right?
Yeah.
So part of the deal is the sacrifice of time, and it's my responsibility to make sure I publish so that their names are in publications, and it makes it easier for them to find residency programs. So right now we're trying to focus on wrapping out all the projects that we have in the lab, and Dr. Josh Weinstein has she's the chief research officer, and she
Hmm.
supervises all of our research. She says specifically told me, do not take new projects on hand, do not. And so I'm very careful. So I will be very interested in looking at, it's COVID obviously. The virus has an effect on the nerves. We all know
Mm-hmm.
that, the olfactory nerve. So would it have an effect on the optic nerve?
Right.
My suspicion it probably does.
Yeah.
And there's some reports of problems with vision, but I have not. And also, uh, you know, working with pathogens is tricky.
Yeah.
So I try to avoid it. It limits a lot the amount of work that you can do.
Mm-hmm.
And for us, not all of our equipment is in the animal house, so we have to bring them up and down.
Yeah.
We use an elevator, and so which it will be difficult to work with pathogens because of the equipment that we have in different rooms,
Right.
and
We
moving
have to
them
keep
around.
everybody safe.
Public space, so safety is very important. So look, uh, I think it's a fascinating topic. I think it's a very good question, but I think we're gonna pass on that one for now, just because of the logistical aspects of it, and also the pressure on wrapping up the projects that we
Mm-hmm.
have. I need to get those done
Sure.
before
Absolutely. You know,
before I leave.
I completely understand that, but I given the current climate that we're in with COVID, I feel like I almost have to ask that question now of every single researcher that I come in contact with, because it's number one. I actually got uh the I guess it was a benefit. um I had the opportunity to talk with my brother on a live stream of his, because he was doing Facebook lives for his church community, and I got to talk about the basic science involved in immunology and what is going on with this virus. And one of the biggest things that I had to tell people was, listen, science is not a quick process. So you know uh the fact, number one, the fact that we got a vaccine at the speed that we did is is fantastic. Again, I don't think I've stressed it enough to people, but 18 months for a vaccine from a virus that we had no idea existed is phenomenal. I can't stress that enough to people. The speed that we moved, and again, it was called Operation Warp Speed here in the U.S., and that's what it was. We were moving at a speed that I don't think vaccine development has hit ever. And I was very impressed at how a lot of the researchers who were working on it stayed quiet about probably all the potential failures that they encountered, and they just carried themselves like you would expect idealistic scientists to operate. They just went to work, they did their job, and they went home, and they didn't go blabbering about potential failures, potential successes. They kept themselves laser focused, and I thought that that was a really good thing that they did.
I completely agree with you. So Joe, you know, uh, you know, I on hindsight, um,
Mm.
I wish everybody was like you in the ability to communicate science. Uh, my wife is not a scientist, but she likes science. I talked to her a lot about it. And one of the things she says is, you scientists need to learn how to communicate better.
Yeah.
You need to be able to explain your science in a way that people can understand it. And the scientific community succeeded at science, which is what we're good at,
Mm-hmm.
but failed at communicate. And
Yes.
if we don't understand that, we will never be able to reach out the people that are skeptical of science. There's a part of us that didn't do a good job.
Yeah.
We did not do a good job at communicating science. We didn't do a good job at explaining to people that were skeptical.
Mm-hmm.
And it's not just the job that you do. You have to make sure that people feel safe.
Yeah.
And also, this is a joke, and of course, I don't mean it, but maybe if we had taken a little bit longer, more-
No,
people would have been
I
more
know
ready
what
to
you mean.
take it because they would have seen more death around. When you see it's hard to accept something unless you see a lot of people surrounding you dying from it. Sometimes.
No,
Um,
you're right.
I'm glad we didn't. Of course, that is a joke, but as a social experiment, it would have been interesting. I
Mm-hmm.
think people would have been more ready to see the vaccine if it had been developed in 2 or 3 years, because they would have seen the catastrophic effects that they would have had, and
Right.
they would be more ready to take something. Um,
Exactly.
that they would have had somebody who's close to them that had died,
Hmm.
or had suffered from bad consequences of it. So I think we need to learn. We need to learn from our mistakes. Scientifically, it was an enormous achievement. I completely agree with you. Intensive communication was a disaster. Was a
Yeah.
disaster that we cannot blame anybody but us. And blaming people for not understanding something that they might have not, they don't know nothing about,
Yeah.
is not a good way. It's not a good way to go. It cuts the communication and it ostracizes them, and it's just not a good way. So we need to learn from our mistakes, and that's what science is.
No,
You fail.
you're absolutely right. That's kind of all a part of the game that we play. You know, we need to learn from our mistakes. We need to learn from our failures. And I say this to students all the time that get frustrated over a experiment that doesn't work, or a theory that kind of gets blown out of the water. Is don't be mad, be better. And that's kind of what we have to do as scientists. Um, one of the hardest experiences that I had as a young scientist, I'm still a young scientist, as a undergraduate, is when I wrote my first poster. My professor put it up, and she had me give a presentation in front of my class, and then she told the class, she pointed to my poster and to me, and she said, "Okay, now I want all of you to destroy it." And at the time when it was going on, I didn't understand what she was doing. But what she was trying to do was she was trying to remove my emotion, my knee jerk reaction to defending my research, and to be more objective. But also, what she wanted me to do was she wanted me to learn exactly what you said. I have to be able to communicate properly so that people can understand exactly what it is that I'm trying to do. And if I fail at that, I failed in part of the process of being a scientist.
Yeah, because as scientists, we have progressed a lot.
Hmm.
So years scientifically, we have progressed very little in terms of communication.
Yeah.
Um, and this that's why I'm so thankful that you gave me the opportunity of this, and of course you take this on yourself, and
Thank you.
you taking the responsibility. I think it's enormously important. Um, I think we're not gonna go anywhere if people don't understand what we're doing. We have to learn to explain and educate. We have to be more humble too.
Hmm.
We have to understand that not everybody's at the same level, and that the things take time to understand.
Yeah.
We have to be approachable, and just not don't judge people so quickly. People have fears. Their
Yeah.
families, their health, is something that is difficult. It's difficult people to accept. So there are many lessons to learn from this pandemic.
Yeah.
And one is definitely that one. And I think communication is something that should be taught um,
Hmm.
in most postgraduate programs for PhDs,
I agree.
scientific communities. I think it should be a must. That and writing. There's, in my opinion, not enough emphasis on writing
Yeah.
when you're an undergrad, and not writing just something, but writing grants.
Yep.
Writing Scientific writing is very different to literature.
Hmm.
You have to have a way of drafting things. Students are terrific, but they come to my lab, and they mix the interaction with the discussion.
Hmm.
Um, they put discussion in the results. They really struggle to
Yeah.
to do scientific writing. And these are people that are highly educated, and they come from several levels of education, and always surprises me, that
Yeah.
and it's not their fault, they just have never been taught.
Well, I mean,
So that
another problem with that can be that, uh, still, to even to this day, there are certain scientific articles that have results, discussion, and then conclusions, all in the same paper, and that makes it very difficult, especially for Americans, to understand, because you've probably noticed this in your time here. I've noticed it pretty much my entire life. Americans love absolutes. We love binary answers. We don't like the the grays, the shades of gray, which is pretty much everything that science is. I find every time someone asks me a question, I go, well, not necessarily. It's somewhat like that, yes and no.
That is very true, Joe. That's very true.
So we're getting close to 7 o'clock, and I promised you that I wouldn't keep you too long because, again, this is your personal time, and I do want to respect that. We have a couple of questions left for you, but I want to give our audience an opportunity to ask any final questions that they may have. So while they're doing that, do you have any questions that you would like to ask me at this time? Anything that's kind of come up to you?
For me or
Yes.
the audience.
Well, for me, that any questions for me that you
What
may
what
have?
what, what led you to create this? I think it's a terrific idea. But there's people with ideas and there's people that bring them forward. I mean, you were in a community of people that I assume you the thing that you got together in common was video games, right?
Yes.
Um, so what drove you to
So,
spend your time to do this?
last summer, my wife, who is a graphic designer and works in the publication field, her and a friend of mine, who is a brand manager, they said to me, your channel's name sucks. And at the time it was a word and it was numbers, and she said your channel name sucks. You need to fix your channel name because people are just not gonna find it. So I said, okay, well then what should I do here? Don't just leave me with that. You got to give me something that I can work with here. So they said, okay, well, you're a scientist, so why not just call yourself Joe the Science Bro and make it somewhat of a rhyme? People are going to understand it easier, and you're just going to have an easier time doing things. I said, oh, okay, fine. And I did that, and I started noticing that people were asking me some scientific questions. So I said, okay, well, let me field some questions. And I started noticing as I kept getting more and more, like almost these tangents of science, I finally said to myself, okay, maybe there is something here that I need to do to properly communicate, at least from my perspective, science. And you know uh I have one student in my lab that I work in. She is a she's pursuing internal medicine, so she wants to be, the main doctor that you see in a hospital. And I said, here's the thing that you're going to have to deal with, and you better get this down pat. You need to be able to explain everything in terms that a layperson is going to be able to understand. So you better get yourself a toolbox and an infinite library of metaphors that you can just pull down out of nowhere to explain things, because you're going to have to explain things to people who have absolutely no idea what is going on inside the human body. And you have to relate that concept to something that they're going to understand. And not every person understands cars, not every person understands golf, not every person understands anything. So it's very important to have a good, like you said, good communication is kind of the hard part that we as scientists have. One of the things that we discussed with Dr. Balin was that anybody really has access to all of our work. Uh, Thankfully, with the way that the internet is and the way that publications work, it's out there for the public to see. But it's written in a jargon and a way that it almost makes it I can't go and I can't read a article on heart muscle and cardiac tissue because it's written in a language and jargon that's specific to that community of scientists, which again is good because it means that there's a shorthand for it, but it's also bad because now I have to overly explain a concept to someone that is outside of my scientific field, which I
Mhm.
think we've progressed so much that we've come up with new terms to describe things, but in a way we've almost regressed in our way to communicate that information.
Yeah, yeah.
So, one of the first questions is a little more general in science. Do patents restrict your ability to do research?
Um, Once you publish uh, a discovery, you cannot patent it.
Hmm.
Um, so you have to patent it before you publish it. Um, Patents are expensive, so they don't limit my research, but they limit the potential capacity of becoming rich, which is very unlikely.
Mm-hmm.
um, so, you know, do I hold results back because there might be a chance of patenting or getting a patent over something? Very rarely have I done that. And when
Mm-hmm.
I have, it wasn't my decision, it was somebody above me.
Okay.
Scientists don't get most of the royalties that come from a patent. The university has
Mm-hmm.
the first call. And so we all have bosses. I have bosses. And
Yeah.
occasionally my bosses have told me, before you publish this, hold it back. We might want to think about whether we're going to patent it.
Yeah.
So, you know, um, yeah.
The
Good
next the last question that we have is why is it that UV light is harmful to our vision, but infrared light isn't?
It has to do with the wavelengths
Mm,
can cause damage to proteins and DNA. Uh, for the same reason that not you can be under your light, a desk, a lamp in your house, and you don't get sunburn.
mm,
But you go outside without sunscreen, and you will get, if you have fair skin, you get sunburn.
right.
Um, Different wavelengths have different effects, and in the case of UV light, it's particularly damaging to DNA and proteins.
Mm-hmm.
The the bonds that form the DNA molecules and the bonds that bind proteins together, some of those bonds are very sensitive to that wavelength. So it has to do with the wavelength of it. Some of it is damaging, and some of it is not. Yeah,
Mm,
but the best analogy I can find is it's the sun versus a lamp on your desk, or just a light bulb that will not be on those frequencies, will not give you sunburn.
okay. Um, I think that is all the questions that I have left over. Um, we did have someone who left the stream early, but she wanted to say thank you for coming on, and she hopes to see you again. If I may, on behalf of my community and my audience, we would love to have you back at some point to have a second conversation with us at your leisure, of course. But yeah, thank you so much for joining us. It has been an absolute pleasure, Dr. Bravo.
This has been a pleasure for me. You made me feel really comfortable. So did your audience.
Thank you.
They're very appreciative and very nice. The questions were terrific, and this has been a wonderful experience. I do this anytime, anytime. It has been wonderful. So thank you for organizing this. Thank you to all of you for coming to these chats and learning more about science and making all those terrific questions.
Mhm.
Um, if somebody of the audience has any questions that they want to ask privately, feel free to give them my email address. I'll be very happy
Absolutely.
to answer them. And I hope to talk to you again. And thank you for this opportunity. It has been terrific. Thank you.
Of course, Dr. Bravo. I would love to offer you the floor here at the for the audience to say anything that you want to talk about. Whether there's a topic that you wanted to discuss that we didn't touch on just yet, or if there's something that you would like to say about the research that you're currently doing, or if there's just any final thoughts that you would like to convey to us. I offer the floor to you right now.
I just want to thank you. I just want to thank you all for your interest in science. We, for being so patient. I know it's difficult to listen so late to a guy with a thick accent, so I really appreciate that. And I just want to thank you all for being here. Thank you. I think what you're doing is fantastic. You're a great communicator and a great
Thank you.
scientist, and it's wonderful to get to know somebody a little bit more. Somebody who is so close to you in physically, but you don't
Yes.
interact as much. So I'm super grateful for this opportunity, and I really, yeah, thank you all. Thank you.
Well, again, thank you so much, Dr. Bravo, for this. Again, thank you so much for offering your time, and please extend my gratitude to your lovely wife and the rest of your family for
I'll do that.
allowing us this time. We really do appreciate it, especially myself. So thank you very much, and yeah, just We really appreciate you, and we hope that you will join us again at some point, whether it's next week or next month or next year. We hope that you'll join us again.
It would be my pleasure. Anytime.
Thank you. Thank you so
Thank you.
much.
Thank you.
All right, let me just do my outro, and then we will all be able to go home and enjoy our lovely dinner. So I want to thank everyone for joining us tonight. I hope you guys had fun. I know that we did. If you enjoyed this stream, please consider give us a follow, as it helps the channel grow. If you're already following, please consider joining our Discord, as that is the best way that you can interact with us. Find out our schedule for things. Find out if we're having a surprise stream, or if there's questions that you would like to ask. That is one of the best ways for you to join to ask me that question. If you're watching this on YouTube, I hope that you will please like, comment, and subscribe, as well as share this video and this content so that we will know exactly what you like. The links are going to be in the description below and on my Twitch homepage. So long, everybody, and thank you for all the fish. Bye bye.